This is the first of 6 video segments in which we will discuss the diversity of ion channels expressed in neurons and how this diversity affects the electrical behavior in different types of neurons. There are many hundreds of ion channels that are expressed in neurons and in other cells in the body, and these channels are thought to have evolved from a single or perhaps a few very simple ion channels that appeared very early in the development of life on the Earth. These simple ion channels were thought to be sensitive to the metabolic state of cells, sensing the ratio of ATP to ADP. These channels most likely had 2 membrane-spanning domains, as simple channels still do today. And these simple channels evolved into more complex channels, having more complex properties that were controlled by various intracellular messengers, including cyclic nucleotides and G proteins. Somewhat later in the evolution of life, more complex channels evolved that were sensitive to the transmembrane voltage. And these channels expressed additional transmembrane domains, one of which acted as the voltage sensor, and this was an alpha helix that spanned the membrane that had several positively charged amino acids that would sense the transmembrane voltage and actually move as the voltage changed. Additional transmembrane domains were added to these channels to shield the positively charged alpha helix, which acted as the voltage sensor. The structure of several voltage-gated ion channels is shown in this slide. What we see here is the structure of an inwardly rectifying potassium channel subunit. This is a very simple channel and probably resembles some of the earliest channels that evolved. This contains 2 membrane-spanning domains, M1 and M2. These are alpha helices, and in between these is a P region which forms the pore of the channel. This is a beta sheet which penetrates most of the way through the membrane and forms the pore. Now this is a subunit of the inwardly rectifying potassium channel. You need 4 of these to form a complete inwardly rectifying channel. A somewhat more complex channel is shown here. This represents the structure of a delayed rectifier potassium channel, and the 2 membrane-spanning domains that were present in the inwardly rectifying channel are still present. These are labeled the S5 and S6, and as you can see, in between them is this beta sheet here, which is the pore region. It's a little complicated, but it's called the H5 region in some potassium channels. But in addition to this structure, which resembles the inwardly rectifying channel, there are 4 additional membrane-spanning alpha helices, and one of these, the S4 region, contains positively charged amino acids and functions as the voltage sensor. And once again, you need 4 of these subunits to form a complete potassium channel. A more complex channel structure is seen in the sodium channel. This is shown here, and as you can see, the potassium channel, the voltage-gated potassium channel up here is represented by this region here. There are 6 membrane-spanning domains, including the S4 region, which is shown in red. That's the voltage sensor, and the P region, which functions as the pore, lines the pore of the channel. But there are actually 4 repeats of this potassium channel motif coded for in just 1 gene, and so all you need is 1 of these proteins to form the entire channel. You don't need 4 subunits, as is true in potassium channels. And if we were to look at this sodium channel from the outside of a cell looking down on the membrane, it would look something like this. Here are the P regions. There are 4 P regions. As you can see, they form the channel pore, and there are also 4 S4 regions, which are the voltage sensors. During this lecture, we'll be surveying the wide variety of channels that are expressed in neurons, and we'll begin our survey by talking about voltage-gated sodium channels. And there's relatively little diversity among sodium channels. There are perhaps 10 different varieties. There are 10 genes or so that express different variants of the sodium channel, but as we'll see, this is a small number compared to sodium or potassium channels. There are both TTX sensitive and TTX resistant types of sodium channels. The classical fast inactivating channels that we talked about before are TTX sensitive. These are the channels that generate the rising phase of the action potential. And even among these fast inactivating channels, there are certain variants coded for by different genes. They have different activation voltages and different kinetics as well. And this is shown in the voltage clamp data right here. In A, we have a series of voltage clamp records showing activation of the current and then inactivation of the current. And as you can see, if you compare this voltage clamp data with that shown in B, the activation and inactivation of the channels in B is quite a bit slower than that in A. So the kinetics of these two types of sodium channels are somewhat different. The activation voltage at which the channel opens is also different, and this is clearly shown in this current voltage plot, this IV plot, which shows that channel A, the channels shown in A, activate at a slightly more hyperpolarized voltage than the channels in B. So we'll see this as a general rule. Voltage-gated channels can vary in a number of ways. They can be sensitive to different molecules that can block the channel. They can have different activation relations where they open up at different voltages, and they can also have different kinetics. Some may have faster kinetics and some slow kinetics. There is one type of sodium channel that differs quite dramatically from the fast inactivating channels that we have been talking about. This is a persistent sodium channel. The voltage clamp data is shown here, and as you can see, this channel doesn't inactivate, or in fact the inactivation is extremely slow. In addition, it activates at very negative voltages, that's shown here. And it is insensitive to TTX. The gene that codes for this channel is NAV 1.9 gene, and this channel, in addition to being TTX insensitive, inactivates slowly and incompletely. One type of classical channel, in fact this is the channel we saw in panel A, is NAV 1.3. These are channels that are expressed in embryonic neurons, and interestingly in the peripheral nerves, if these nerves are damaged, these sodium channels are upregulated again. And because these channels activate at a more negative voltage, they actually increase the excitability of neurons, and in fact are responsible for increased excitability and pain that's generated in damaged peripheral nerves.